Good, good afternoon, everyone. My name is Pakavarin Khunphet. I am a student from Prince Songkhla University. Today, I would like to present in the topic of olfactory oscillation in response to social ability tests in female mice. First of all, I would like to talk about the background in this study. Humans, as well as gregarious animals that live together in society, it is crucial to express the appropriate social behavior with different people in social life due to increasing survival and reproductive fitness. On another hand, impaired social ability was found associated with various kinds of neuropsychiatric disorders, including autism, schizophrenia, depression, and anxiety disorder. Moreover, social recognition is used as a generic term for both the ability of a subject to categorize conspecific into different classes such as sex and familiarity. How to examine social behavior? In humans, we can talk or ask for social behavior, right? However, most social behavior studies have been focused by using animal models. In the figure, you can see animals communicate by smelling, which is a common social behavior in rodents. Moreover, according to previous study, women are more likely to be diagnosed with psychiatric disorder rather than men. However, most social behavior studies in animals have also conducted solely using male animals. So, female animals were used to test the social ability in this study. Most social behavior had highly focused only on behavior tests. However, the evidence from previous studies have implicated that several brain areas regulating social behavior, including olfactory bulb, the hippocampus, and the nucleus accumbens. Moreover, rodents use olfactory information for smell. Therefore, the olfactory bulb is mainly known as the first relay center in the olfactory neural pathway, displayed by the olfactory cue input for social information in the rodent. LRP recording was used to study the activity of the olfactory bulb in this study. What is local field potential or LRP technique? LRP technique is a recording of the electrical activity generated by the neuron in the brain area. If we record the neural activity of the olfactory bulb, so we can analyze the LRP pattern that might represent the involvement in social behavior. So this study aimed to identify and characterize the neural oscillatory patterns related with social recognition in normal female mice by using LRP signal from the olfactory bulb, the main social associated brain area in rodents. Okay, this figure shows the experimental design. Female swift ICR mice were used in this study. Electrodes were implanted into a olfactory bulb. After animal recovery, LFP signal and animal behavior was recorded together during animal perform a social ability test. Next, I will expand about electrode implantation. All animals were anesthetized by intraperitoneal pre-injections of psilocin and followed by Zoretil. The surgery was started after animal deeply anesthetized. Electrodes were implanted into olfactory bulb according to Malbane atlas. Animals were administered antibiotic ampicillin to prevent infections and allow them to recover for at least 7 to 14 days. The social ability test. The sociability test was used to examine social behavior. The test composed of key phases, including baseline, habituation, and social ability tests. In the baseline section, animals were placed in the empty chamber and let them explore for 5 minutes. 20 minutes later, animals also placed in the same chamber, and two identical cups were also placed. Let animals explore for 5 minutes. In social ability tests, same as message mouse was used as a stimulus mouse. The stimulus mouse were placed in the one side of the chamber and let testing mouse explore for 5 minutes. The behavioral parameter was automatically scored by optimal program. LFP signals were also recorded during animal performed social ability tests. As shown in the figure, this is raw data from olfactory bulb, but this signal did not tell the LFP pattern. So Pakavarin transform used to transform the raw signal into power spectrum with different frequency range. The frequency range were divided into specific bands, including delta theta, alpha, low beta, high beta, low gamma, and high gamma. So why we divided the frequency range into specific bands? According to previous study, shows specific frequency range show a change in the behavior. Next this is a result from behavioral test. Figure A is show the habituation. This bar graph represents the total time spent of animal in each zone. Zone 1 is empty cup and zone 2 also empty cup. You can see here animals spend time not different between zone 1 and zone 2. However, in figure B, it shows the social ability test. This bar graph also represents the total time spent of animal in each zone. Zone 1 is social target and zone 2 is empty cup. You can see here animals spend more time in social target comparison to empty cup. Next, this picture shows the raw LFP signal and spectrogram during animal perform the baseline, habitations, and sociability tests. The color code of spectrogram indicates the power of signal. Red represents high power and blue represents the low power. The spectrogram shows an increase of high gamma power, especially during a sociability test. However, these LFP signals were transformed into a power activity. This is the diagram showing power spectrum during baseline, habitation, and social ability test. Y-axis is a power showing a percent total power. X-axis is frequency wave between 1 to 100. And we found the difference in high beta, 19 to 30 Hz, and high gamma, 61 to 100 Hz. So then we average the power within this frequency range and show in the bar graph. For statistical analysis, we found that decreasing in high beta and increasing in high gamma during habituation and social ability tests. Moreover, both high beta and high gamma were significant different compared between habituation and social ability tests. In discussions, our findings demonstrate that female might also display an increase in time spent exploring social targets. According to previous social behavior studies, social recognition tests in rodents depend on the animal's interests, motivation to investigate other individuals. Particularly, rodents are common investigate novel and familiar objects as well as stranger conspecific. The consistent findings were also reporting the previous study, which indicate that female also increased time interactions with a social target in contrast to male mice. Our findings also demonstrate that decrease of high power and increase of high gamma power in habituation and social ability tests comparison to baseline. The olfactory bulb found that beta and gamma band are closely related with odor processing, attention, perception, and cognition. Previous study in male mice which demonstrated an increase of 5 gamma power follow social investigation, while gamma rhythm are more related to local infrastructure processing. That the concept of beta rhythm is an organizing of long-range communication between brain areas. However, the change of beta and high gamma power during social ability were more pronounced and significant difference compared to habituation section. In conclusion, the reduction in power in beta power and enhancement of gamma power in olfactory bulb associated with the response to social stimuli rather than novel object exploration. So, specific response of the olfactory bulb signal may indicate the end course of social exploration. Thank you for your listening. Any questions?
Okay, thank you very much, Ms. Pakavarin. Your presentation is nice. Okay, and to the audience, do you have any question or suggestion to the presenter? Okay, I have some questions. The first, what did your result can be applied for the further study in your idea?
Okay. This result is like normal. It's called like baseline data for. You can study of the effect after some neuropsychiatric disorder. For example, autism or something is, it is associated with impairment of social ability. So this like to be baseline data. If you want to study the impairment of social ability, you can apply more about like medical something.
Okay. To the audience, do you have any question or suggestion? We have the time. Okay. The last question. Why did the sociability test have a decrease in the high beta power when compared with the habituation? Could you interpret this result?
According to previous study, they thought beta power is about like communication with another brain region, but gamma power is like in the local of the neuron. So this time maybe it have some connection between the area, this social brain related area, something. Might this occur to another area, but I did not sure too.
Okay. Thank you, Ms. Pakavarin, for